So what I wanted to cover in these few slides, as kind of that makeup for the fact that we didn't have any sound on Wednesday, is just again going over endospore formation and the very beginnings of biofilm formation. So when we first started the class, we talked about a little bit how even unicellular organisms can sometimes display properties of differentiation. For the most part, they don't. Most unicellular organisms have one particular way that they look, and that's their morphology. That's what they look like, and that's fine. But some do undergo different life stages, different morphologies, different phenotypes, and so that's what this section on endospore formation and biofilm formation are going to kind of be about. So endospore formation occurs under adverse conditions. So it's a lot like a stress response. Really, a lot of the same things that would trigger the stringent response that I just talked about would also trigger endospore formation. However, the monitoring system and the relay system for undergoing endospore formation is significantly more complex. Forming an endospore is a way more permanent way to deal with stress than just doing a stress response, because endospore formation really requires a commitment to the fact that you're going to lose your vegetative capabilities. You're going to be packaging your DNA up in this very, very dense, very thick spore matrix in order to wait out months, years, decades, millennia, millions of years of stress until conditions get better. So the system that endospore forming bacteria have in order to induce sporulation, or the process of forming a spore, is a more complex relay system. It's a lot like a two-component regulatory system or a signal transduction cascade. It's just more complex than a sensor kinase activating a regulator. And again, that's because the cell has to really determine that this is an important thing for it to do. So what happens is you get this phosphorylation cascade. This protein phosphorylates that protein, which phosphorylates that protein, which phosphorylates on and on and on, until eventually you are phosphorylating this protein called SPO-A, a sporulation factor, a protein involved in spore formation. Once SPO-A is highly phosphorylated, that's when the cell really has the signal that says, okay, we're going for it, we're going to form a spore. Now, this slide has more details on it than I really need you to know, but what we're trying to see here is that external signals for sporulation, desiccation, cell density, starvation, whatever stress response it is, you get this cascade of phosphorylation, that will eventually phosphorylate SPO-A, which continues to activate different sporulation factors and will eventually lead to spore formation. Okay. So what else happens during spore formation? Well, the cells have to form this really complex, really thick, really dense structure to protect themselves. And if we're talking about a condition where the cells are stressed out, usually because of a lack of nutrition, where in the world are they going to get the chemicals and the products they need to make this complex structure? Well, as you can see on this slide, they get those components from cannibalizing other cells. What will happen when that SPO-A protein is highly phosphorylated? You get the release of a toxin. So that toxin will kill neighboring bacillus, allowing the cell that produced the toxin to consume the bacillus. The cell that produced the toxin will also produce an antitoxin to protect themselves. So at the same time that this cell that has a highly phosphorylated SPO-A is secreting all these toxins and killing all their neighbors, they're also producing a protein that delays sporulation in other systems. So they're killing their neighbors, and if they don't kill their neighbors, they're at least going to prevent them from making a spore. And so those cells that have entered sporulation first are the ones that will be successful because they will use the neighbors that they've killed for nutrients, and they will be the ones that will form the endospore. Usually under those nutrient limiting conditions, especially when cells start to run out of phosphate, because remember this is a complex phosphorylation cascade. When they start to run out of phosphate, they'll increase the transcription of that toxin gene to, again, make sure they can kill the neighboring cells, get as much phosphate as possible so that they can still continue their phosphorylation cascade. And then finally, I'll introduce biofilm formation, even though we talked about biofilm formation on Friday as well. Biofilms are this really important structure. They're cells embedded within a matrix, and there are 4 basic stages to forming a biofilm. They have to attach to a surface. Once they've attached, they'll colonize, meaning they'll begin to secrete a polysaccharide matrix. The cells will reproduce. Then you have the stage called development, where the biofilm gets bigger. It can form these really complex, really large 3-dimensional structures. And then finally, at some point, some of the cells within the biofilm will disperse, or they'll move on to a new environment. Attachment is mediated just by a random collision. A cell will bump into a surface, and if it has a structure like a flagellum or a pilus, that can help it attach. There are other surface proteins that cells can use to help them attach as well. Once they do attach, that triggers some gene regulation within the cell that will turn off motility-related genes and turn on genes necessary for secreting the polysaccharide, continuing to adhere, etc. And then at some point, again, cells will decide to disperse. And so we picked up on Friday with this image, so I'll go ahead and end this makeup of Wednesday on this image.